Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Tariq O'Dali from Business Review, and I will be your host. It is our pleasure to have Syngene International Limited with us today, who will be presenting this webinar titled Oligonucleotide Pharmaceutical Development. Today's guest speaker is Ramesh Rajeshwaran, Head Oligonucleotide Pharmaceutical Development. You're going to be hearing from him in a moment, but first I'd like to welcome you to our webinar platform. Now this is On24, and you'll notice this webinar is browser-based. So if you do disconnect for any reason, just click on the link you received via email to immediately rejoin the session. In order to ask questions, you can send them in at any time via the questions widget. So that's the box at the top left-hand corner of your screen. Don't forget to click the submit button to submit your question. We plan to allocate some time at the end of today's session to address any questions or thoughts that you may have for us. Please use the yellow help widget at the bottom of your screen if you require any assistance, and you can also move, resize, and maximize any of the windows in front of you to get a better view of the slides if you need it. So if you need to go full screen on the slides, you can do that as well. But now, housekeeping out of the way, please allow me to welcome today's speaker, Ramesh. Over to you.
Thank you, Tarek. Good morning and good afternoon or good evening, depending on wherever you are in any part of the world. Welcome to the webinar on the topic Process and Analytical Development of Therapeutic RNA Oligonucleotides Systematic Approach. In this presentation, I intend to cover the agenda, as you can see on the screen. The overview about Syngene, Syngene as a company, and the oligo capabilities at Syngene. And the main topic of the presentation of the process development and through a structured approach by the way of design of experiments, and the analytical development that goes hand in hand with the process development of a oligonucleotide. And to illustrate the structured study, we'll also go through a detailed case study along with the data that we generated. So, with that brief outline, now we move on to the overview about Syngene. So, what is Syngene? Who we are? What do we do? Syngene established itself as a company in the year 1994. That's about a quarter of a century earlier. And since then, we've been doing some great work and having some unparalleled experience. It's been a great journey. Initially, we started as a discovery services. Primarily, we are a CRO company. Discovery services in the fields of small molecules and biologics. And later, the gamut has been expanded to cover discovery, development, and recently clinical development, as well as manufacturing services for small molecules and biologics. Syngene has one of the largest talent pools comprising of various different disciplinary, multidisciplinary scientific teams, numbering over 3,000, and you can find some of the top talents working at Syngene. We are based out of Bangalore, India, and you can see a snapshot, an aerial snapshot of the facility in the slide. We have several audits by major life sciences partners across the world. Our facilities get routinely audited. We have been audited or certified by most of the leading quality agencies. We have a GLP certified animal facility. Our facilities have been inspected by the FDA for the manufacturing as well as the quality systems. And our phase 1 clinical and bioanalytical labs have been audited by FDA, EMA as well. In terms of data integrity and IP protection. We have an impeccable track record since our inception, so much so that there is no single instance of any breach of confidentiality. Currently, in terms of business, Syngene is a publicly traded company in the Indian stock exchanges. Our current market cap is over $1.5 billion. So, having set the stage with who Syngene is or what Syngene does as a CRO, I switch over to the topic for the evening or for the morning, the therapeutic oligonucleotides. I don't need to talk about oligonucleotides to this audience. Nevertheless, oligonucleotides are short DNA or RNA fragments, usually up to 25 nucleotides and sometimes more, which could be chemically synthesized using nucleosidic monomers. Some of the subclasses of therapeutic oligonucleotides based on their mode of action are antisense, siRNA, aptamers, and most recently the exon skipping and CRISPR-Cas based mode of actions. On the right hand side, I just have a brief cartoon where the DNA undergoes the transcription to the mRNA and which further translates to the protein in the disease causing mechanism where the protein causes the disease. How does the antisense work? Actually, the antisense avoids the mRNA to translate it into the disease-causing protein. So this is one of the mechanism or mode of action in the therapeutic oligonucleotides. Similarly, the other classes follow the mechanism depending on what the therapeutic subclass is. The oligonucleotides are applied for various therapeutic areas like oncology, vaccines, CNS, or other biotechnology applications. And in terms of the commercial or the approved drugs by the FDA, we have in fact 5 drugs, starting from Vitrovin, which was approved way back in the 90s, and the latest one is Spinraza, which the oligonucleotide fraternity knows well about. So, having set the stage on the Syngene presence as a CRO as well as the therapeutic oligonucleotides, I'll just briefly explain Syngene's foray into the oligonucleotide business. Why oligonucleotide? A few years back, when we evaluated our growth path at Syngene, one of the growth engines, or we evaluated various technologies, and among these technologies, one of the growth engines was oligonucleotide technology. Once this is identified as oligonucleotide or a new growth engine, a new technology area, then we set about creating this into a business as a function at Syngene. Syngene is known for its expertise in setting up new capability that we have developed over the past 25 years, and leveraging this expertise, we had this set up in terms of the process development and the pilot scale-up, which I'll talk in a while. Also, for any new technology area, the expertise is important, and the team of experts comprising of some of the brightest talents in the oligonucleotide space have been set up with a mix of PhDs and engineers. And we understand that any new technology area needs a good amount of hand-holding. So we work with some of the leading consultants in the oligonucleotide area in setting up the equipment, validating them, training our people, and troubleshooting as and when it is required. So with these levers, we have successfully set up the oligonucleotide business unit at Syngene, and currently that actually makes a good segue into my next slide, which talks about the capability development at Syngene. Like I told a few years back, to be precise, the end of 2013, a process development facility was set up with the OP400, and the full train of the upstream and the downstream capabilities were set up. This facility is currently running, and we have run several projects on the process development and the small scale synthesis on a non-GMP. And the scales that we operate typically are from 250 micromole to 8 millimole. Having been in this field for quite some time now, it was quite clear for us that the future is in the scale-up. And towards the end of 2014 or early 2015, we set ourselves the goal of setting up a CGMP facility on the OP400 scale. By the beginning of the last year, this facility was set up, and the facility was qualified as per the GMP norms. And this facility also was employed to release the first clinical batch towards the end of the last year. This facility can handle the scales from 4 millimole to about 45 millimole, and it is a completely QA monitored GMP facility. So what's the future? The future lies in the scale-up and the process scales. So we have set ourselves the sights on the oligo process scale in probably a couple of years from now, and this plan is at a conceptual stage at Syngene. So this runs through the capability that we developed over a period of time, and currently the oligonucleotide facility at Syngene, we can confidently tell that we handle most of the natural, unnatural, modified oligonucleotides, oligoconjugates, or the cholesterol-based conjugates, the lipid-based conjugates, etc. And we can handle that at a scale of the 250 micromole to 8 millimole in the process development on a small scale, and from 4 millimole to 45 millimole in the scale-up facility, the GMP facility. Currently, this is a facility which is available for our clients and with readily available capacity. So, I'll change gears and go to the main topic of the discussion of this slide, of the presentation, which is the process and the analytical development. So, once a molecule is discovered, then it needs to undergo a process development. But why a process development, and how a process development is done? So once a molecule is past its discovery phase, it needs to be advanced through the development cycle, as it is the case with any of the pharmaceutical developments, and it needs to be handled efficiently, made efficiently for the talks and the clinical studies. The drug substance need to have a very good quality as well as an efficient way to produce it. To achieve the high quality, a systematic process development approach is very much necessary. This should also, without any question, be handled or supported with a very good analytical development, which normally goes hand in hand with the process development. In the following slides, we will attempt to describe our approach on these aspects. And complementing the process development is an important aspect, which is the control strategy. The control strategy per se is all the parameters which have an impact on the final product in terms of the quality as well as the yield. For example, the starting materials play a very important role. The starting materials, that is the raw materials that's used in the process, like the phosphoramidites, the solvents, the reagents, everything plays a very critical role in the efficiency of the oligonucleotide manufacture. The phosphoramides in particular needs to be controlled very well in terms of its purity as well as its impurity profile. For example, the phosphoramides need to be controlled to a purity greater than 98% with all the impurities much under control. So how do we do it? We have a very stringent quality measure and in-house check on any of the incoming raw materials. We have worked with our suppliers to actually establish the quality at the receipt stage so that we do not see any surprises right from the development stage up to the scale-up. Likewise, the phosphoramidite needs to be controlled for the amine impurities, which could play a very critical role in the synthesis. The residual amine needs to be checked thoroughly using a proton NMR, which is done in-house as a routine analysis. Likewise, the solvents like the acetonitrile, etc., play a very critical role in terms of its moisture content, and we work with our clients to ensure that we get the material of the right quality. Excuse me. And the next parameter and the most important, or one of the most important, is a very critical in-process monitoring of the process at various stages. The in-process monitoring needs to be employed to ensure that the process is on track. For example, at the end of the cleavage and deproduction in a typical oligosynthesis, the crude purity is determined. The pH is determined at various stages of the process by the way of strict controls. We have ensured that these control strategies are in place to not to impact the product in an adverse manner. Talking about oligosynthesis, it is purely a automated synthesis based on equipment, and a good level of the equipment control and the monitoring is paramount for the control of the process. The equipment consists of various parts which needs to be maintained well. The regular maintenance, calibration of the equipment, as well as, for example, checking the flow rates prior to every synthesis. These are all the controls, and including the spare parts, the valves, the pipes, the O-rings, and the seals, etc., need to be in place to have a very efficient synthesis. Moving on to another important parameter, the material handling and the storage. The final oligo, we all know, is or could be temperature sensitive. Routinely, this is stored at -20 degrees, but depending on the modifications on the oligo, the temperature could vary. If it is a DNA-based oligonucleotides, it could be, it need not be very stringent because it is more stable compared to the RNA. Nevertheless, the controls need to be in place to ensure that it doesn't impact the quality in any adverse manner. Likewise, any material that comes into contact with the final product needs to be controlled by the way of handling sterile materials as well as autoclave materials. So on the whole, this is a control strategy that we have tried to establish on the process as well as the product. So now moving on, the next slide talks about the process, typical process cycle, right from the synthesis and the cleavage and deproduction up to the downstream. A typical oligosynthesis involves solid synthesis on a solid support. It starts with the detritiation, coupling of the phosphoramide, followed by the oxidation or the thiolation depending on the backbone, and capping, and then the cycle repeats itself. So we all know that this is a typical synthesis cycle, and there are several factors which impact the efficient synthesis. So at this point, there are several factors that can be studied, and structured DOE, a design of experiment, could be in place. For example, solid support and the detritiation or the coupling and the oxidation reagent. So in just a moment, I will talk in more detail about the design of experiments and the parameters or the factors. But on this next step, the C&D, the cleavage and deprotection, which forms an important part of the process from the upstream, where a structured DOE can be performed. And also in the downstream, in the purification and the ultrafiltration, there are several factors that impact efficient purification and ultrafiltration, and which have a scope to be studied in great detail. So that sets up the tone for the next slide, where each of the parameters are touched upon in slightly more detail, and how a structured DOE can be formed around this. Not all molecules need to undergo extensive DOE. It depends on the modification or the complexity of the molecule, as well as in which phase the particular molecule is in. So depending on the early or the late stages, structured DOE could be adopted. As a strategy, we have performed DOE on some of the simpler oligonucleotides, which will be actually applied to any new incoming molecule, and the process or the DOE modified as per the requirements or as per the complexities, the modifications involved. Like for example, in the first parameter, the solid support, there are various factors in the solid support, like the type of the solid support. This type of the solid support plays a very critical role. Different supports are available commercially. For example, it could be polymeric based or non-polymeric, like the CPG, the conventional CPG or the hybrid CPG. In terms of the polymeric support, the kind of support is chosen depending on the kind of backbone. Phosphatidyl backbone could be done on a nitophase, and GE unilinker, for example, on a phosphodiester backbone. So depending on the complexity, this could be decided. And CPG, for example, presents a very economic option, as it has several advantages. It does not swell. It's available with the different porosities, etc. So depending on the kind of development that is needed, as well as the phase in which, as well as the complexity of the molecule, the factors could be picked for a detailed DOE. Another important factor is the loading of the solid support. It varies in terms of the CPG. It could vary anywhere from 40 to 100 micromole per gram, depending on the kind of molecule that is the oligo that is studied. The next parameter is the synthesis. The synthesis involves two critical steps: the detritiylation and the coupling in the synthesis cycle. In case of the detritiylation, depending on whether the oligo is a DNA or RNA, the acidic concentration of the D block reagent is chosen. Generally, dichloroacetic acid with varying percentage is used. Likewise, the coupling is affected by the activator, and different type of activators could be employed for the coupling using varying concentrations. Routinely, the activators like ETT, BTT, or DCI is used with different concentrations. Some of the other important factors are actually the ones which impact the contact time of the reagents on the solid support. These could be controlled by studying the various flow rates as well as the recycle time. So we have quite a few factors on hand which could be put together to form a structured DOE. The next step in the synthesis cycle is the oxidation or the thiolation, as I told earlier, depending on the backbone. So for the oxidation, routinely the iodine is employed as a reagent in a mixture of pyridine water. For the thiolation, pads or a EDIT or a Bucage reagent could be employed. And the concentration of these reagents play a critical role, and these are studied in great detail depending on the molecule. Another important factor is the viscosity of the reagent. Due to the viscosity, there could be back pressure issues in the synthesis, and these need to be adjusted and are done routinely using some solvents like acetonitrile. So these are the various parameters that are studied in terms of the for the oxidation parameter. Another important and critical stage in the synthesis is the cleavage and deprotection on the upstream. The type of reagents actually vary depending on the linkages on the solid support. The solid support usually comes with a succinyl linker, and which are base labile. So generally bases like ammonium hydroxide or the ammonia methylamine solution is used for the CND. These come in a range of 25 to 35% as a commercial material, and the concentration can be played around with, and also the proportion of the reagents can be varied to have an efficient CND process. The CND process is very important because the maximum amount of the oligo that is formed needs to be cleaved out from the solid support, which has an impact on the final yield. So the temperature and the time of the CND is studied in detail. We employ a temperature range of about 20 to 70 degrees to study these factors in the form of a structured DOE. So having talked about the upstream process, the other important process, or the other important step in the process is the purification and the ultrafiltration. In terms of the purification are done routinely using quaternary ammonium-based strong ion exchange resins like the GE source Q or the TOSO Q. The bead size of this play a very crucial role. About 15 to 40 micron sizes are available, and these are studied with respect to different oligos. The column radius plays a very critical role as well. Different buffers could be studied, for example, tris buffers, phosphate buffers, sodium hydroxide, sodium chloride based buffers, etc. And of course, without mentioning the elution parameters play a very important role, because the right fraction need to be cut off from the during the purification to enhance the product quality. On the ultrafiltration, the more important or the most important factor is the transmembrane pressure, which in turn impacts the flux. That's also studied in detail. So in terms of the factors, as we can see, there are several factors which impact the efficient synthesis as well as the downstream process, and these could be studied in a very structured way. So I now move on to the next slide, where we talk about the scalability. The DOE helps us to fix the parameters which works well for a particular oligo, and we all know that the oligosynthesis is automated synthesis and equipment based, and there are several factors in the scale up that needs to be studied well in the process development. So for this, typically we use a scale down, scale up model, depending on the capabilities on the scale up at the OP400, which will ensure a facile scale up to happen. The synthesis bed height, for example, or the loading capability of the OP400 is actually calculated theoretically. Based on that, the experiments are planned on the small scale, the micromole or the millimole scale, using small scale SS reactors, adjustable columns, the fine line 70, etc., the small scale. So these are studied in great detail, and the scale-up factors are fixed at the OP400, and which actually helps to develop a process and scale it up without any risk to the product or to the final quality of the product, and get it in a good yield. Likewise, the robustness of the process also is improved depending on how the scale-down, scale-up model is employed. In the next slide, the similar process or the similar methodology is used for the other important step in the oligo manufacture, which is the purification. The column efficiency is of paramount importance for purification. The packing of the column determines the column efficiency, and if the column is not packed well, it leads to issues like channeling, etc. during the scale-up. This is studied experimentally, and using calculations, the asymmetry, which is a factor of the column efficiency, is studied before the scale-up. The asymmetry is typically in the range of about 1 to 1.8, and the case here illustrated shows a very good packing efficiency, which is also observed in the peak that's illustrated here. The flow rate forms a very important part of the purification as well, and this is where the scale-down, scale-up model works beautifully for the good scale-up. The fine line 200, which is the capability at the OP400, OP400 facility is studied for the bed height that is possible using a constant linear flow rate. These are then calculated at the downscale and studied at the micromole and the millimole scale using columns like the high scale 26 or high scale 50, where the process is scaled up at 2 times, and then from the high scale 50 to the fine line 200, it is a 4 times scale up. The parameters for the scale up are fixed at the PRD level so that it does not lead to any surprises, and the synthesis is taken up efficiently in a robust way. So that actually forms a very good detailing about the process development and the DOE factors that needs to be studied. So at this time, I'll just take a pause and hand it over to Tarek for a quick poll.
Thank you very much. Yes, that's right. So now it's time for an interactive poll. So audience members, please get involved. What you can do is you can vote on one of these 3 options you have in front of you here and hit submit. So the question is, a detailed DOE study would be a helpful tool in the development of an oligonucleotide-based API. And we'd like you to respond, please. So if you agree, say agree, disagree, or don't know, that's the 3rd option there. Don't forget to hit submit to submit your vote. We're going to leave that on the screen for about a minute now, and while you make your selections, I'm just going to hand back to Ramesh now to briefly talk a bit more about the question today. Ramesh?
Yeah, sure. Thanks, Tarek. As I explained in the talk, in the previous slides, a detailed DOE study is an efficient or a helpful tool for any development on the oligonucleotide. As you could have seen, there are various factors which determines the purity and the final outcome of a good synthesis, and a detailed DOE study, if not a detailed, at least for the important factor, would be really helpful in determining the path forward for any oligonucleotide.
Lovely. Thank you very much. Just going to give the audience a couple more seconds here. A detailed DOE study would be a helpful tool in the development of an oligonucleotide-based API. Agree, disagree, or don't know? Please select your answers now and hit submit. Just a couple more seconds. And that's it. Thank you very much to everybody who took part in this poll. And now let's have a look at the results. And okay, so it looks like the majority of the audience there have gone for A, agree. So is that a surprising result for you, Ramesh?
No, not really, Tarek. Any process development chemist or any oligonucleotide chemist would definitely agree with this, seeing the number of factors actually that impact efficient synthesis. Okay, fine. So now that we are done with the poll, I will quickly move on to the next topic, which is the analytical development. So any process development needs to be hand in hand working with the analytical development, because the process doesn't have any meaning without the proper analytics. So that actually forms a good base for the next slide, where we talk about the analytical method development strategy. As we all know, oligonucleotide contains sugar, heterocyclic base, phosphate, which all add up to the complexity in a molecule, and which makes a method development all the more complex. Added to this, most of us know that there are no prescribed or definitive guidelines from a regulatory agency for the analyticals. So in the absence of this, the analytical method development needs to be a very solid strategy. That's where we start with a platform method, which we have developed over the past few years, and apply this to any new oligo that we have in hand. Coupled with a good literature search, once this is the platform method is applied, we see if the method works well for the oligo, and depending on the method efficiency, this is further optimized in terms of the parameters like pH, the diluent, organic modifiers, the gradient, as well as the stationary phase. Oligonucleotides need to be analyzed using specific stationary phase like a DNA pack 100 or a DNA pack 200, the OST columns, which have a very good impact on the way the method is developed. Also of importance is not only the main peak, but also the impurity. The impurity identification and characterization is one of the important strategies that needs to be developed as a part of the method development. In terms of the method, once it is developed, we perform a pre-validation activity to study its LOD, LOQ, precision, linearity, etc., and ensure that the method is a robust one. And finally, this method is transferred to the QC to have a control over the process during the scale-up. So the next couple of slides, I just talk about the orthogonal methods, the HPLC method, basically the ion exchange and the ion pair method. It's important to have orthogonal methods, as I told earlier, in the absence of analytical specification guidelines. It helps to have orthogonal methods to be employed. Based on our studies for this particular illustrated one that is in the slide here, the sodium phosphate buffer works well, and the method is further tweaked in terms of the pH, the purity profile based on the pH. For this study, at the various different pH, the peak shapes are somewhat distorted. In some of the cases, the impurities are not well separated. So based on this, the pH 10 has been chosen for this particular method. Likewise, on the ion pair method, the ion pair method is a very handy one because it's quite sensitive as well as has a good resolution. A classic ion pairing agent, the HFIP, the hexafluoroisopropanol, is used, and in this illustration, the method is studied at different temperatures from 65 to 70. At 70 degrees for this particular method, there is a good separation of the N-1, N-2 impurities, as well as a good the resolution of the impurities, as well as a good separation of the FLP from the impurities. So the smaller impurities play a critical role, and as can be seen, the method development is a very critical part of the plays a critical role in the development cycle. Okay, so that forms the initial part of the talk, and now in the next few minutes I'll take you through a case study where we studied the double-stranded siRNA. The sequence is just illustrated. It is a typical 18-mer oligo of the sense and the antisense strand, which is then finally annealed to get a double-stranded siRNA. These are made of natural amidites, and the scope was to actually develop a robust process and scale it up in the cGMP facility. The plan was to perform the batches on a 1 millimole scale and scale up from the lab to pilot scale at a 10 millimole scale. Now I'll move on to the next slide, which is it talks about the synthesis. So, like I talked about in the DOE, for the smaller scale, the micromole and the 1 millimole scale, the DOE was performed to find the exact coupling and the oxidation condition, and then this process was transferred on to the scale up. So in this graph, actually the plots depict the detritylation curve, which is a theoretical indication of the progress of the synthesis. The green line indicates the 1 millimole synthesis, and the red one indicates the 10 millimole synthesis. So the curve shows that the synthesis is reproducible from the small scale to the large scale, and the synthesis progress in a similar course for both the scales for the sense strand. Likewise, the same is experienced for the antisense strand. So, an efficient DOE and a scale-up study at the small scale has resulted in a good and efficient synthesis for the 10 millimole scale. Likewise, for the other part of the upstream, the C&D, which as I mentioned, is a very critical process. The process flow has been illustrated on the left side of the slide. The conditions were decided based on a DOE. Ammonium hydroxide methylamine 1:1 solution was used, and the C&D process was followed. And at the end of the C&D, the ultrafiltration was performed to remove the deprotected protecting groups, and the pH was monitored to the to control the ultrafiltration process. At the end of the ultrafiltration, you can see that the sense strand has a OD of 1.3 million, and the antisense strand has a OD of 1.4 million, which roughly translates to an yield of about 65 to 70% at the crude stage. The purity of the crude is greater than 80. It's about 80% in both the strands. So this shows that the synthesis and the C&D up to this stage has proceeded quite well on the 10 millimole synthesis. Now moving on to the next critical stage of the ion exchange purification. So the ion exchange purification is performed using prisma as a buffer, and the graphs, the plots shown here depicts the chromatogram obtained from the purification. The broad peak represents the major product, and during the purification in the ion exchange, the main fractions are collected from the broad peak and analyzed for the purity of each of the fractions. The next slide gives the details about the analytical details of the fractions. The absorbance is on the extreme left, and the quantity of the product in terms of the OD as well as the purity of the FLP in terms of the HPLC percentage. A good look at the data shows that the fractions from 5 to 11 has the highest quantity of the product in terms of OD as well as the purity. These fractions are then taken to the process called as the mock pooling, and the which I will talk in a while. And the mock pooling decides the way forward for the purification. So I'll just quickly move on to the next slide, which talks about a similar analytical of the fractions from the Antisense strand. And here again, the major product lies in the fractions from 8 to 11. The other fractions showing very less product or less purity. So these fractions were collected, the fractions from 8 to 16, and taken for mock pooling. So what is a mock pool or a pooling decision? The mock pool is basically where the various fractions are collected and analyzed for its yield as well as the purity in a small scale, and then the decision to pool the larger amount of the fraction is taken. This is typically a time-consuming process, and during this process, the material is stored under lower temperature, typically -20 or lower. This decision is in consultation with the client, where the purity needed and the specifications are fixed, as well as the yield is determined. If the higher purity is desired, then it might result in a lower yield. So it becomes a very critical decision. So in this case, the mock pool 1 represents the best in terms of the yield as well as the purity. Likewise, the similar process is followed for the Antisense strand, and the mock pool 1 once again results in a high yield as well as a desired purity of 90%. At the end of the purification, the ultrafiltration is performed to salting out the to control the sodium by the desalting. The pH is maintained to near neutral, and the conductivity is monitored to control the sodium counter ion, thereby having a good control of the sodium in the final product. The HPLC purity is determined at the end of this process, which in this instance is greater than 90%, and shows that the yield as well as the purity is good. The final process is the annealing, where both the strands are annealed to get the double-stranded siRNA. This process is typically monitored or done on a lot-wise addition of one strand to the other, depending on the concentration, and done over an interval of time, and is studied using the SEZ. Over a period of 150 minutes, we can see that the single strands gradually disappear and the duplex formation happens at the end of 150 minutes. So once the product is annealed, then it is taken for the freeze drying process, and at the end of this, a lyophilized product is obtained as a fluffy white colored solid, which in this 10 millimole scale was about 60 grams, which roughly translates to 50% yield of the purified product. This translates to a very good and efficient synthesis of the duplex siRNA. The last slide talks about a full release analysis of the siRNA. I will not go through each of the lines, but the analytical details need to be very comprehensive, and we have we check all the parameters in-house with our own capabilities. So with that, I come to the summary of the presentation. We have attempted to describe identification of critical quality attributes for efficient process development, design of the experiments for a structured synthesis, and demonstrate it in a small scale at the 1 millimole in the lab, and successfully transferring that to a 10 millimole synthesis to achieve the desired purity and the yield. So with that, I just show a brief snapshot of the facilities at Syngene on the process development scale as well as on the OP400 scale under the GMP. So with this, I come to the end of the session, end of my talk, and thank you for your attention, and the session is open for any questions.
Brilliant. Thank you very much. Just a reminder for the audience, in order to ask your questions, you can send them in via the questions widget. So just type them into the box there at the top left-hand corner of your screen and click the submit button. We're going to try and take a couple of questions now before we wrap things up today. And this first question we have here. So this person says, how do you demonstrate analytical comparability between API made at OP100 and OP400 scale?
Yeah, yeah, that's a very good question. As I explained in my slides earlier, we do a very detailed method verification exercise as well as a validation. The validation consists of studying the LOD, LOQ, the linearity, as well as establishing the robustness. And this actually ensures that the method is transferred from the small scale to the large scale. Also, as a CMO who've been involved in the method transfers for a very long time, we have detailed SOPs in place where the method is transferred from the analytical lab to the quality control in terms of the study of the validation parameters. So that's how we ensure the analytical comparability between the OP100 and the OP400 scale. Tarek.
Thank you very much. Next question we're going to move on to now. This person says, when you have a process optimized at OP100 and want to scale up to OP400, how do you define contact times for the OP400 scale?
Yeah. Once again, the process optimized at the OP100, we define it based on the flow rates, and the flow rate determine the contact time of the solid support during the detritylation, coupling, and the oxidation. It also depends on the diameter of the column that is studied. And overall we study the flow rates to ensure that the process that is optimized is scaled up at the OP400.
Okay, thanks a lot. Questions. We've got some great questions here. Audience members, keep the great questions coming in if you want to have your question answered. We're going to try and get to a couple more here. This next question is, what kind of solid supports can be used for a typical 30-mer oligonucleotide API? Yeah.
The kind of solid supports that should be employed for an oligonucleotide depends on the oligonucleotide itself. For example, whether it is a DNA or a RNA, and also it depends on the length of the oligo chain. For a 30-mer, typically a polystyrene support with about 300 to 350 micromole per gram, or a CPG with about 70 to 90 micromole per gram can be used. But as mentioned, we do have some base experiments performed on typical oligonucleotides, and this could be extended to any kind of oligonucleotides that comes our way, starting from the baseline that we have already established.
Thank you. Thank you very much. Next question is, so this person asks, can UPLC or HPLC ion pair method be transferred from instruments of one make to another make?
Yes, most definitely. It can be done, but it needs further optimization or modification. It depends on the make and model of the instrument from one to which it is to be transferred. It also depends on the method, whether it is transferred from the HPLC to the UPLC or vice versa. For example, water and Dionix UPLCs have different wide volumes due to difference in mixer capacity. So the conditions need to be optimized or tweaked when it is transferred from one make to the another.
Thank you very much for the answer there. This next question, so this person says, what is the minimum and maximum synthesis capability at Syngene for typical DNA or RNA oligonucleotides?
Yeah, for a process development and the small scale synthesis, the capability is in terms of quantity, it's about 300 milligrams to 15 grams. On the micromole on the molar scale, it is 200 micromole to 5 millimole. On the scale up, which is a GMP facility, the synthesis scale is from 5 millimole right up to 45 millimole, which could roughly translate to about 15 to 140 grams in case of single strands using PS support. For the CPG support, it could vary depending on the loading, and it depends from one oligo to the other. Tarek.
Thank you very much. We're going to try to get to maybe one or two more here before we wrap things up today. This next question is, what was the reason to detect iron, nickel, magnesium, etc. in API?
Right. If you see a typical specification of an oligo, as per the guidelines for a clinical batch, the extent of the metal content needs to be determined. But it purely depends on the development phase in which it is, and that is exactly the reason that we have analyzed these metals in the product that we synthesized. And also to highlight another point, we do have the capability of determining the metal content using some of the high-end analytical techniques in-house at Syngene. Tarek.
Thank you very much. We're going to try to get to one more very quickly. This next question is, do you perform forced degradation studies as part of control strategy for impurity profiling?
Yes, the simple answer is yes, we do forced degradation studies as part of the control strategy.
Okay, lovely. And I think that does bring us to the end of our time here today. So sorry to everyone whose questions we didn't get to, and I'm afraid we are all out of time. So that just leaves me to thank today's speaker, Ramesh, for what was a great presentation. I'm sure you'll all agree. And of course, to thank Syngene International Limited for sponsoring this session. To the attendees, you will receive an email very shortly, and that will tell you how you can access the on-demand version of the webinar if you'd like to catch up or if you've missed any, or you can access it on our website. The website is www.business-review-webinars.com. We do look forward to sharing further webinars with you in the future, so please keep an eye out on that website. You can also follow us on Twitter at BRWebinars for daily updates, and of course join our LinkedIn group, Business Review Webinars, as well to stay up to date. Once this webinar ends, today's survey will appear in its place. We would appreciate it if you could spend a few moments just answering the questions in there for feedback. It's very important to us, your feedback, so we do appreciate you staying to do that. Thank you all once again, and I hope you all have a lovely day.